Hello, my fellow doctors and nurses friends. Welcome to the other episode of Lab Rat Sherry. Today, let's chat about why the lab asks for more blood than the test catalog told you. Before we start, make sure you subscribe and hit the like button. So first, the dead volume. This does not apply to every single lab, every single analyzer. It's more frequently to happen in a smaller hospital at the rural area with a relatively older model. Some of the analyzers, to ensure the test sampling probe does not going all the way down to the bottom of the test tube and crush the bottom of the test tube, they will have a fixed position of how far this sampling probe will travel to pick up the sample. And that safety measurement is the reason you will have dead volume in a test tube. There will be some liquid at the bottom of the test tube, cannot be picked up and sampled. Next is the repeat and dilutions. So the minimum volume on a test catalog means for one try, not for series dilutions or repeat of the test. So here is what I mean. If you have a patient with very high glucose or very high HCG or CRP or something like that, what happens in the lab is we will put on the analyzer for one run, and then we will receive a flag saying, oh, this is outside of the testing range and we need to start to do dilutions. And because nobody really knows how high the number really is, what happens is maybe one dilution is not enough, and then the sample ends up needing multiple dilutions and multiple repeats. That will ending up with a much higher volume need than what the test catalog say. And unfortunately, not every single lab allows series dilutions. That requires much lesser volume than individual dilutions. What I mean by that is a series dilutions means you make one dilutions for say 1 to 2, and then you can use that 1 to 2 dilution to make 1 to 4 and then 1 to 8. But some laboratories, they don't allow that happens. They only allow to make 1 to 2 dilutions, and then you restart fresh to use the neat sample to make a 1 to 3 dilution, and then use the neat sample again to do 1 to 5 dilution. And that type of requirement will ending up with even higher volume requirements for the sample. Third, the aliquot. Especially in the chemistry department, a lot of tests, they cannot be all on one big analyzer. We might actually need 2, 3, or even 4 or 5 of different analyzers to finish everything you ordered. So what happened to them is, in order to give you a result as fast as we can, and also make sure the sample doesn't expire before we finish all the testing, we need to aliquot them. And every single aliquoted tube, they need a little bit extra volume for the potential dilutions, repeat, or dead space. And that will be another reason why we're asking much more volume than what you see on the test catalog, even after you adding every single test requirement up. And last, let's talk about the ones that confuse so many doctors and nurses so much is the sample requirement for whole blood versus serum. Hematology department is the only department doesn't have these confusions because they are using the whole blood for testing. But for every single other department, coag, blood bank, or chemistry, these departments are using serum or plasma to run the test. But a lot of times, the minimum volume on the test catalog, they are giving you the volume based on the spun down serum or plasma, not the whole blood. If you see some tests that requires 200 microliters or 0.2 ml of sample, it actually means approximately of half ml of whole blood. And after we spun down, they will be left in relatively 300 microliters or 0.3 milliliters of serum or plasma for testing. This situation goes especially worse if we are having baby samples. The little human in this side, they have a really high hematocrit. If we use procalcitonin as an example, what happens to this sample requirement is you will go to the test catalog and see a requirement saying to run a procalcitonin regardless of adult or babies, you need 0.2 milliliters of blood. But that 0.2 milliliters of blood for testing means 0.2 milliliters of serum or plasma for testing. A lot of babies have a hematocrit of over 50, and now the next thing happen is When you pull out a capillary tube and getting a heel stick, that capillary tube holds the maximum of 600 microliters or 0.6 milliliters of whole blood. After we centrifuge that blood, it leaves us less than 0.3 milliliters of serum or plasma for testing. The other part is packed red cells. They are not suitable for testing the procalcitonin that you want. So for this type of sample, even you feel like you give us a full-filled capillary tube that holds 600 microliters of blood or 0.6 milliliters of blood, we can actually only run it once on the analyzer. Fortunately, a lot of times this is not a test that need a repeat, but if this is something else that actually need a repeat, this is the time you will get a call from the lab saying we don't have enough blood. So now, here is the conclusion. Sometimes when you call, what you might only want to know is the bare minimum of giving one try. But when we output the answer, we actually played a whole season of drama in our head and try to tell you the minimum volume of ensure an answer encounters all the possible plot twists. That is what caused the mismatch of the volume you see on the test catalog versus the volume we say in the phone from an actual bench tech. Thank you for watching. This is Lab Rat Sherry. I'll see you in the next episode. Bye!